Hey guys, welcome to AP Biology Gene to Lecture Series, lecture number 4. We're going to be talking about mutations today. And Shaggy, problems do happen every time your cells divide. Remember, as I said earlier, every time your cells divide, you have approximately 30 new mutations in your cells, which is actually incredibly accurate, but they still do happen. And again, just to reiterate, once again, the molecule of DNA is an incredible molecule because it allows for the faithful reproduction from generation to generation, but also allows for change, i.e., through mutation. The problems, or I should say, the sometimes benefit of mutability. Okay, so there's several different types of mutations that can occur. One of the most common type of mutation is a point mutation. It's just simply a single base pair change where a one letter of the code gets changed for a different letter of the code. Now, as I said earlier, during replication, DNA replication, mutations are relatively rare. Again, because DNA polymerase acts as a proofreader. Now, that does not occur during the process of transcription, where we're going from DNA to RNA, and RNA polymerase is reading the DNA template strand in order to make that pre-messenger RNA molecule. There is no proofreading ability in that RNA polymerase, and so mistakes are made about every 10,000 base pairs, which is pretty high considering many of the genes are 10,000 base pairs long or longer. Okay, a lot are also shorter, but many are pretty quite large. And when you get a point mutation, there's basically three typical outcomes: what we call a silent mutation, a missense mutation, and a nonsense mutation. Silent mutation simply is where a change in the genetic code does not cause a change in the amino acid sequence. And remember, that is due to the redundancy of the messenger RNA codon, of the codons that code for the amino acids. If you remember, leucine has 6 different codons that do code for the amino acid leucine. All right, so oftentimes we can see a change, and oftentimes that change is going to be in the third nucleotide of a codon. Okay, and that's oftentimes what we will call the wobble effect. There's just simply no change in that. In the amino acid sequence. Okay, now missense is where you get a change in the nucleotide sequence that does lead to a change in the amino acid sequence. And once again, the severity of that particular change depends upon what amino acid was substituted. If we substitute one polar amino acid with another polar amino acid, similar properties, then oftentimes there may not be much of a functional change in that particular protein. However, if a polar amino acid is substituted with a non-polar amino acid, then that may lead to a significant protein shape change, which could lead to a significant increase or loss of function of that particular protein. And then finally, the most severe type would be a nonsense um, uh, mutation, where all of a sudden what was once an amino acid coding codon now is turned into a stop codon, and production of that protein is terminated early. And again, so how can the question on there on the bottom says, how can mutations affect the next generation? So think about that. Where must mutations occur in order for them to affect the next generation? I'll let you answer that question. All right. Now, the other type of mutation, which is very severe, is called a reading frame mutation, where everything behind that mutation is going to be changed. And the way that happens is usually through insertion or deletion. Is probably the most common way, where a particular nucleotide is actually deleted from that from the messenger RNA molecule, or even in the DNA, or another nucleotide is inserted in place. Okay, so we get an addition there. And as you can see, what we're starting off with is our top frame, and we would have up here, we have methionine, lysine, phenylalanine, glycine, and stop. Well, here, what we see in the next frame down below, the frame shift causing extensive missense, is that the U that would be at phenylalanine is removed. And so remember, the ribosome is reading 3 nucleotides at a time, one codon. So it's reading these 3, then it's reading these 3. Now it's going to read the next 3, which is going to be U, U, G, because we took that U out. And so that gets changed, the phenylalanine gets changed to leucine, and now we're reading a different series. Behind everything is changed. Okay. The other one is where a frame shift causes immediate nonsense, okay, i.e., we get a stop codon is created all of a sudden and stops production of that protein, and that can be pretty bad. Now, generally, if 3 or multiples of 3 nucleotides are removed, oftentimes that may not be as bad because it may just remove a couple of nucleotides and it may not cause that much of a change in the overall shape of that protein. Of course, it could potentially cause enough of a change that that could cause problems or maybe even be a benefit to the organism. It all depends upon what happens to that particular protein and how the environment, i.e. natural selection, reacts to that particular. Does the organism survive? Does it survive better? Does it produce less young? Does it produce more young? All of that is going to be based on, is going to affect the outcome of that particular mutation. So as an example, here is a simple sentence composed of 3-letter words, and we're going to read that as the fat red cat ate the rat. Okay, that's my sentence. All right. Now, just think about it, if I took out, let's say we just removed the word fat, okay, and then we have the red cat ate the rat. Okay, we just remove this word fat and we'd have the red cat ate the rat. That still makes sense. Maybe that, and I can still understand what's going on there. So if we lose 3 of those nucleotides, it may not be all that big of a difference. But if I just remove the F here, now what's going to happen, we're going to frame shift and we're going to read that as still 3 letters at a time, but it's not going to make any sense. Now we have the atter, itic, atta, tet, at. Does not make any sense at all. And that would be an example of a frame shift mutation where the ribosome is now reading a different set of 3 nucleotides at a time. Okay, now you might go, well, what's the problem with losing one nucleotide? Well, or changing one nucleotide? Well, sickle cell anemia is a great example of that. So what we see up on the top, or I should say on the left-hand side, is we see the wild-type hemoglobin DNA. Okay, and sickle cell the hemoglobin gene, I think, is 100 and I can't remember if it's 126 amino acids long or if it's 126 codons. I think it's 126 amino acids long. Okay, but somewhere like around the 4th or 5th, there is this point mutation at position 6. Okay, that would be the 6th nucleotide, which would be the second, the 6th nucleotide, which would be like the second codon. We have a change. Instead of a thymine, we have an adenine. So what does that do? The messenger RNA, instead of being GAA, which codes for glutamine or glutamate, we now have CAT, which now codes for GUA, which now codes for the amino acid valine. And again, glutamic acid is hydrophilic, valine is hydrophobic. And that simple amino acid change is going to make that hemoglobin sort of sticky. Okay, and what does that mean? That means during periods of low oxygen, the hemoglobin molecules are actually going to clump together into a long chain, which is going to make the red blood cells form a sickle shape, which now they become rigid and they cannot pass through the capillaries, and they have a tendency to block the capillaries up like a bunch of logs in a small stream. And blood cannot get to the extremities, cannot get to organs and things, and leads to cell and tissue death and extreme pain. Okay, there is a single base change that leads to a lethal disease if it's not treated medically. Okay, and of course, sickle cell anemia is a autosomal recessive trait. So both parents, if they're carriers, they have a 1 in 4 chance of having a child that does have sickle cell. Now, remember, these are just probabilities. There's a probability that, parents have 2 kids and both of them have sickle cell. What's the probability of that happening? That'd be 1/4 times 1/4, or equal to 1/16. And so the fact that this couple had 2 kids with sickle cell in a row would just be bad luck. Okay, and of course, we all know the story of sickle cell. It is a disorder that we find predominantly in areas where malaria is dominant, or prevalent, I should say, and the having sickle cell, or not having a sickle cell, but being a carrier for sickle cell, having one copy of that sickle cell gene, does infer a certain amount of immunity to dying from malaria. And malaria is probably one of the biggest killers in the world of kids. And again, if somebody asks me what is the most dangerous animal in the world, I would have to say it's the mosquito, mainly because of malaria, dengue, yellow fever, chikungunya. There's all sorts of mosquito viral vectors, born viral vectors. So there's lots of them. Okay. Now, we just learned in the intro phenomena that we had for this particular unit that 2 gene therapies have just recently been approved by the FDA, which is going to involve the use of CRISPR-Cas9. Do notice the date on this article, December 8th, 2023. So this is very recent. But again, frame shift mutations. This is an interesting example. This is an 11 nucleotide deletion in exon 3 of the myostatin gene that inhibits muscle growth. So we have this gene called myostatin that, when it's active, it inhibits muscle growth, which is good because then we don't get all so bulky and things of that nature. Okay? And do notice it's a protein produced in myocytes, muscle cells, that act via an autocrine pathway to inhibit muscle growth. Remember what autocrine? That's self-signaling. That's a cell giving signals to itself to prevent it from dividing. And here's an example of this dog right here where that mutation has occurred. And I don't know about you, but I would not want to meet that dog on a dark alley. And here's a picture of a bull that also has that particular mutation. Where is that mutation found? It is found on chromosome number 2, and it's found on the 2q. The q means it is on the long arm of the chromosome. So that would be, this is the long arm, because here's my centromere. This is the P arm, which is petite, and here is the Q arm right here, which is the long one. And it's between base pairs 190,055,700 to 190,062,729 on chromosome number 2. It is over 7,000 nucleotides long, and 11 nucleotides were deleted to lead to that outcome. Okay, so that's just a kind of a cool little picture of how we, and this has been found in humans. Actually, it was first discovered in an Austrian family where all these kids were coming out, as soon as they were born, they were like bench pressing their mother and stuff. A hugely muscular family, first found in humans and then found in other animals. So that's why we have a picture of a human chromosomes here. And again, do you notice this is only one set of the chromosomes? Okay, well, so there we go. All right. Chromosomal mutations. What's the difference? Well, these are large mutations that involve multiple genes and are a result of alterations to the structure of one or more of the chromosomes. So this is the whole chromosome actually is mutated. And so some of the various mutations we're going to see there are inversions, which are going to change the order in which genes are found. And that can really play a havoc if it's on something like what we call the Hox genes. Deletion, where we just take out a whole bunch of chromosomes. Duplication, where we duplicate a section of the chromosome. And translocation is where one chunk of a chromosome gets put onto a different chromosome. So here's duplication. You can see we've duplicated B. Deletion, we've lost B. Inversion, we've put B on the bottom arm versus where and B and E have switched locations. And translocation, we have two different chromosomes here, and we've put part of one chromosome onto another chromosome. As you can see, E and F from this chromosome have now changed places with the other one. So there's been a translocation. All of those are going to I should say duplication, inversion and translocation are going to alter the sequence of genes, which, again, like I said, can have some major outcomes. Deletion, of course, we lose that particular gene. Duplication, on the other hand, can do something interesting because the organism still has a copy of the gene, has two copies now, but now that section of chromosome that has been duplicated is sort of like almost free to basically undergo change without most likely too much of a problem for that organism. And duplication, we now know, has played an important role in the evolution of higher organisms. And we'll get into that in Unit 7. And again, here's just another picture. Again, uh, translocation, inversion, relocation of genetic material. Deletion, loss of genetic material, missing chromosomes. Duplication, gain of genetic material. And again, as you can see, we can duplicate the entire chromosome. Entire genomes can actually even be duplicated in many types, or in many organisms, especially in plants. It doesn't work too well with higher vertebrates at all, but we know it has happened in our evolutionary past. And again, we'll get into that in Unit 7. And that is it for our lecture on mutations. I hope you enjoyed it.